and welcome to today's webinar titled Panel Presentation, Automating High-Throughput Luminex Assays for Multiplex Gene and Protein Analysis. This webinar is part of the 6th Annual Laboratory Automation Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex, a data-storing company. For more information about Luminex, go to luminexcorp.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. I now present today's speaker, Dr. Stefan Jellbauer, a product manager at QuantiGene Gene Expression Assays and Luminex Instruments, and Dr. Bani Suri, a product manager software at Laboratory Automation. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Welcome to both of you. You may now begin your presentation.
Thank you very much for the kind introduction, Susie. As Susie mentioned, we are speaking on behalf of Luminex Corporation today. Both my colleague, Dr. Bani Suri, and myself are employees of Thermo Fisher Scientific, and we are an official partner of Luminex. So welcome to this presentation about automating high-throughput Luminex assays for multiplex gene and protein analysis. We are honored to speak today on behalf of Luminex, a data-storing company here at this event. As mentioned, we are from Thermo Fisher Scientific, an official partner of Luminex. Here you can see Thermo Fisher at a glance. We pride ourselves in being the world leader in serving science, as you can see by these impressive numbers. At our core, we are scientists, and our purpose is to enable our customers to fulfill their scientific purpose. We really take pride in our mission, which is to enable our customers to make the world healthier, cleaner, and safer. And we offer products and solutions that enable our customers to push the boundaries of innovation. Here's today's agenda that I will be presenting together with my colleague, Dr. Bani Suri from our lab automation group. I'll first walk you through some introductions on high throughput screening. Then I'll present to you our in vitrogen ProcartaPlex and QuantiGene Plex assays for multiplex protein and gene expression analysis. And then Bonnie is going to take over and talk to you about our lab automation solutions and also present our solution for automating our Luminex-based assays. Here we'll be focusing on QuantiGene Plex assays for automation. As mentioned, we offer products and solutions that enable our customers to push really the boundaries of innovation. We're pushing these boundaries with our solutions like the ones presented here, in this case, catering to high throughput screening needs. And I will walk you through some of the examples today. The workflow of high throughput screens usually includes 3 main steps. You first have your screening experiment, where you can screen thousands and thousands of compounds depending on the goals that you have with your screening experiment. The next step would be that after the screening is over, you have to prepare these samples for analysis. So there you could be looking at hundreds of thousands of wells and many 96 or 3D4 well plates that you have to prepare for the analysis, which is the third step here as shown on this slide. And here the key takeaway is you want to measure your key biomarkers as outcome of your screening with the goal of finding a lead that works for your pathway or for your condition that you're investigating. So this analysis can be either for RNA, proteins, metabolites, or other omics parameters. As part of the analysis, a data analysis tool and platform is also needed, and this is something that's sometimes overlooked, but I want to stress the importance of this as well. In order to reduce the time to results and to increase the efficiency of your high throughput screen, many labs are looking into automating several steps or the whole workflow, and we'll present some solutions here today to do this with two particular assays. Today, I'd like to present to you high throughput analysis solutions for RNA or protein measurements and automation solutions that we propose for these assays. At Thermo Fisher, we offer in vitro QuantiGene Plex and ProcartaPlex assays that are especially well suited for high throughput multiplex gene expression or protein analysis, as I'll explain to you in the next few slides here. This table that you see here shows a comparison of several specifications for these assays. On the left, the QuantiGene, and on the right, the ProcartaPlex assay. Notably, you can see that you can measure up to 80 genes, so RNAs, and up to 80 proteins with these assays in each well of a 96 or 3D4 well plate. Combined with a simple sample prep requirement, these assays are really perfect tools for high throughput needs. You can run a wide variety of sample types from many different species, depending on your needs. These assays are analyzed and therefore require a Luminex instrument for readout. You can see a suite of Luminex instruments here at the bottom, which, by the way, you can also purchase directly from Thermo Fisher Scientific. And the reason for running these assays on Luminex instruments is because they rely on the well-established so-called Luminex xMAP technology, which stands for multi-analyte profiling, In brief, Luminex invented small magnetic particles or beads that are dyed or that are coated with a different mixture of dyes, which provide a unique fluorescence emission to different beads, and therefore enabling the concept of multiplexing. We at Thermo Fisher are assay developers and take advantage of this principle to build multiplex assays that can analyze up to 80 targets for RNA and 80 targets for proteins. And we use specific oligos or antibodies to capture these targets from various sample matrices. A simplified model for both scenarios is shown here in the center, where we show the antibodies coded to the beads in the center, and to the right, we show the oligos bound to these Luminex beads, which then build the scaffold to capture the targets and then do the signal amplification as with the QuantiGene Plex assay. Notably, Thermo Fisher Scientific is the only supplier to offer both protein and gene expression multiplex assays on the Luminex platform. And on the right-hand side, you can see a nice example of a cell culture stimulation experiment from which the same sample was then analyzed for gene expression using a 36-plex QuantiGene Plex panel, and for protein expression using a 20-plex Th1, Th2 cytokine chemokine panel from our ProcartaPlex suite of assays. This analysis will provide you with answers from many, if not all, your key biomarkers for both RNA and proteins, and I will show you some data for this experiment in a different slide. Thermo Fisher Scientific, as I mentioned, is the only company to offer both protein and gene expression RUO multiplex assays on the Luminex platform. And here I'm showing you some key benefits of both of these assays that are now also available in 3D4 well format for higher throughput demand. The ProcartaPlex and the QuantiGene Plex assays are also tested and validated on the new IntelliPlex platform. You can see an image on the right of this new instrument, and we could show an excellent correlation when running the assay on various Luminex instrument types and comparing it to the results from the IntelliPlex instrument. Some key benefits of the ProcartaPlex assay are shown on the left-hand side. As mentioned, up to 80 proteins from a small volume of sample. We have a lot of different off-the-shelf but also custom panels for this assay, and you get very consistent results. You can scale easily from 96 to 3D4 well plate without losing performance. On the right-hand side, some key benefits of the QuantiGene Plex assay. This is a direct from sample assay, meaning you do not have to isolate or purify the RNA. You go directly from your lysate or you homogenate into the assay, and there's a huge array of different panels available, both custom and now also off-the-shelf panels. Here is an elegant example, as I mentioned earlier, of the concepts of two applications on one Luminex instrument, showing some data of the experiment I mentioned earlier. So in this experiment, U937 cells were stimulated either with PMA or with LPS for 24 or 48 hours, respectively. After the experiment was done, the supernatant was taken off and measured using a ProcartaPlex panel, the Th1, Th2 cytokine chemokine 20-plex panel here. And the cells themselves were lysed and then analyzed on a QuantiGene Plex 36-plex panel for both gene expression and protein analysis. On the right, I'm showing two examples of two targets, IL-8 and MCP1. For these two targets, we found that there's an excellent correlation between the transcript and the expressed protein, as you can see by the unstimulated versus the stimulated condition, where both the RNA and the protein amount or relative amounts are increasing post-stimulation for both targets. Now I'd like to provide you with some details about these Luminex assays, and we'll start with our ProcartaPlex assay for multiplex protein detection. We recently launched, as I mentioned, the 3D4-well format of these assays as well. As a quick recap, on the right-hand side, I'm showing a little scheme of how the Luminex xMAP technology works. Again, you have a set of magnetic microspheres that are color-coded with combinations of red and infrared fluorophores. Each bead set is coded with a reagent specific to a particular bioassay, in this case, a capture antibody, which allows you to capture and detect several specific analytes from a single sample. Lasers in the Luminex instruments then excite the internal dyes that identify either the microsphere, so the bead itself, or the reporter dye, which is captured on the detection antibody, which then provides the quantitation of an individual analyte on each microsphere. So this technology enables you to multiplex the measurement of up to 80 targets in every single well. On the bottom right-hand side, you can see the workflow of the assay. It's a very simple ELISA-like workflow. As mentioned, we recently launched the 3D4-well format of these assays as well, and here is a few key highlights of these assays. They provide very high throughput without compromising the sensitivity. They have a high convenience in terms of the workflow, as mentioned, very ELISA-like workflow. You can scale from 96 to 3D4-well formats with confidence and without losing performance. And in general, they have a very high and robust performance, as exemplified by the R-square values, for example, for looking at the standards. And I'm very excited to let you know that we also launched a new format for even more convenience, which is the lyophilized bead format. This format allows you to directly add your sample to the plate, which comes pre-filled with lyophilized beads. So this is something that we recently launched and is offers you even more convenience to run your high throughput assays. And this is in addition to our standard liquid bead format here, where you would pipette the beads into the plate. Importantly, we also provide a free of charge data analysis solution for these assays, the Invitrogen ProcartaPlex Data Analysis App, which is available on the Thermo Fisher Connect platform. This enables the analysis of the data that come from the ProcartaPlex multiplex protein assays, and these data can be acquired either on any Luminex instrument or also the so-called Bioplex instruments. You can immediately start your biomarker profiling by easily assigning lot-specific ProcartaPlex assay in this data analysis tool to your multiplex data. This intuitive cloud-based analysis tool allows to analyze both 96 and 3D4-well data. You should take a look at the analysis tool here. The link is mentioned on the bottom of this page, and you can easily get publication-ready data out of this tool or for any of your internal or external presentations. For the remainder of this presentation, we'll focus on the QuantiGene Plex assay for multiplex gene expression analysis and showcase automation options for this high-throughput assay. This assay relies on the so-called branched DNA technology, which amplifies the signal and not the target as you do with your traditional qPCR gene expression technology. It is a cost-effective and reliable and scalable method for general gene expression studies, but it also used extensively in high-throughput screenings to measure gene signatures and biomarkers, for example, in drug discovery. Here are some of the key benefits. As mentioned earlier, again, you do not need to purify your RNA. You go right from your lysate or homogenate into the assay, and we offer sample prep kits for both tissues and cells, as well as blood and even FFPE. You can do a multiplexing of up to 80 targets in the 96 or 3D4-well plate, and you can also detect very subtle changes because this is a linear assay. You can also measure percentage differences, not just fold differences. As mentioned, this is available in 3D4-well format. I'm going to show you here a high-throughput cell screening workflow using the QuantiGene Plex 3D4-well assay. Here's a detailed overview of the complete assay workflow, including hands-on and incubation time for day 1 and day 2 of the assay. This assay has an overnight incubation on day 1. As you can see from the number of steps, while hands-on time is fairly minimal, there are various touch points in the workflow that require lab technician time and effort for incubation, washes, pipetting, transfer steps, etc. Here on the left is a simplified view of the different assay steps for day 1 and day 2 that I just showed you in more detail on the previous slide. The adoption of equipment and automated equipment for simplifying this workflow is more and more required by our customers. So on the right-hand side, I'm showing you some required or suggested equipment to run the assay in either 96 or 3D4-well format. You will see that in the 3D4-well format, it's definitely recommended that you use equipment for the different steps, be it for liquid handling or for dispensing, and then definitely for magnetic plate washing. And then you have your instruments listed on the bottom right-hand side that are able to measure either the 96 well plate format or the 3D4-well plates format. So our customers are more and more switching to high throughput applications, and we saw a great opportunity in looking into more automation solutions for this established QuantiGene Plex, as well as the ProcartaPlex assays, by the way. So before we talk about our approaches of automating these assays, I want to show you a couple of published examples of how our customers have already done a semi-automation of these QuantiGene Plex assay and used them for high throughput screening. So in this first example that's on the screen right now, Danahay and colleagues studied cytokine-driven goblet cell metaplasia in the lung, which, by the way, is a hallmark of chronic lung disease and lacks a curative treatment to date. Using the high-throughput QuantiGene Plex 3D4-well assay for compound library screen in 3D bronchospheres, they were able to screen almost 5,000 compounds and found that Notch2 is required for this goblet cell metaplasia in the lung. So more specifically, they concluded that the inhibition of the Notch2 pathway prevents goblet cell metaplasia induced by a broad range of the stimuli that they measured. And they are also now exploring future therapeutic uses in preventing this condition in airway diseases. So in summary, the QuantiGene Plex 3D4-well screen was extremely helpful to reach their goals. In the second example here, Ferrer et al. Used the QuantiGene Plex high-throughput assay for biomarker studies in iPSC-derived retinal pigment cells. They started out by establishing the assay first in the 96-well format and thoroughly validating the assay in that format before they then scaled up the assay to run it in a 3D4-well high-throughput format to increase both their throughput and decrease the required sample input, which was very important for them. The authors found a significant correlation of the multiplex gene expression data from those QuantiGene Plex assays with the standard qPCR measurements, and then they concluded that this high throughput assay provides really the basis for their compound screen to study the retinal pigment epithelium function and maturation, and in exploring future treatments for several retinal degenerative diseases that are out there. So these high throughput studies obviously generate a vast amount of data that needs to be analyzed. So before going into more lab automation solutions, I also want to mention that we have a data analysis tool for our QuantiGene Plex assays as well. And again, I can't overemphasize that data analysis is a big step in the workflow and cannot be overlooked. So our free of charge QuantiGene Plex data analysis software, which is also available on the Thermo Fisher Connect platform, lets you analyze your data from gene expression with an easy-to-use workflow that provides customization capabilities for example, normalization and your general data analysis needs for your multiplex data. So this data that comes from the QuantiGene Plex assays can be analyzed in this software, and then after you're done with the basic analysis in this software, they can even be exported into the so-called transcriptome analysis console software that some of you might be familiar with from microarray analysis. And there we enable the generation of heat maps, PCA, and volcano plots, or looking into pathway analysis. Some of the key features are mentioned at in the table at the bottom of this slide. And by the way, I want to remind you that we have the similar analysis tool also for our ProcartaPlex assays. So when looking at options on how to automate certain steps and ultimately the whole workflow for Luminex-based assays, we looked no further than our Thermo Fisher Lab Automation Group, which has tons of experience with automating various assays and whole workflows, as you can see in this overview slide. So here we're looking from manual all the way to very high throughput capacity systems from left to right. Obviously, you're going to increase your time to results. You're going to decrease your time to result, and obviously the data that you can measure increases with higher throughput capacities. So with this, I will hand it over to my colleague from our lab automation group, Dr. Bani Suri, for the remainder of the presentation. Thank you very much.
Thank you, Stefan. Hi, everyone. I am Bani, part of the lab automation team. So to help everyone understand where lab automation fits in the expansive portfolio of Thermo Fisher Scientific, we were acquired over 20 years ago. And since then, we have a dedicated business within Thermo and have since then formed active collaborations and connections throughout Thermo Scientific. We can boast of the largest install base of over 2,600 installations worldwide over the past 25 years. And this has been possible as we have a dedicated R&D application scientist team, as well as sales and service support, partnering and providing personalized solutions for all our customers' needs. The lab automation team is based in Burlington, Ontario, Canada, and in Langenselbold, Germany. And we also have a global presence where we have substantially expanded our local resource and support infrastructure worldwide, particularly in Europe and in Asia. So when thinking about automating your lab, people often thought of it as a high throughput screening tool. But we and our customers can assure you that automation goes well beyond increasing throughput. So starting with the most obvious benefit, automation reduces scientists' time on manual tasks, such that It frees up time to allow you to focus on higher cognitive things that robots don't do well. Well, at least not yet. And this obviously results in maximizing throughput and instrument uptime, such that your work hour is still your workday is still an 8-hour workday, but the experiments and workflows run 24 hours or even over the weekend. And automation also reduces errors, thereby providing reproducible quality results which are publication ready. With the flexibility to scale and connect data for traceability, creativity would be the only limiting factor for your automation platform. In here, I would like to give a brief overview of what a typical automation system looks like and our current capabilities. So starting at the bottom, which is the base layer is made up of automatable devices. We offer a series of automated incubators called Cytomat and are capable to integrate over 450 instruments, which are automatable, thermo and non-thermo devices. We remain brand agnostic. As long as the instrument's automatable, we can work with it. The middle or the second layer consists of our configurable automation. This includes robotic movers, where we offer stationary movers, which could be stationary or they could be on a linear track. We also have tall movers gaining space in the vertical axis. We also have industrial arms, which is preferably chosen if the payload is higher and the work is intensive. We also have smart technology, which is touch-enabled, such that when you touch the handles of our smart shelves or smart carts, they respond by disengaging the devices from the automation platform, allowing their safe use in an offline manner or if they need to be transported. And the top, or the brain controlling it all, these layers, is the Thermo Scientific Momentum Workflow Scheduling Software, or Momentum for short. So Momentum allows communication across and between all these layers, and also between cloud and external repositories such as LIMS. The core value to our portfolio, as you can see, is collaboration and flexibility. We like to think of automation as a journey, and in our experience, we've come across 4 journeys that our customers have been through. The very first on the left is the instrument loading journey, and that is when someone is looking to automate a simple instrument loading and unloading setup for their workflow. It's just to displace the manual loading and unloading of 1 or 2 instruments. As we move from left to right, the requirements get more complex and the systems get more advanced. So as we move to the simple workflow, the second one, the customer is typically looking to automate a larger assay or a slightly bigger workflow with maybe 3 or 4 automatable instruments. Moving further right, which is the configurable platform journey, here we are looking for a more configurable and flexible solution to connect more processes and adapt to changing workflows in the lab. This is what we are going to be focusing on for the QuantiGene Plex assay. And then there's also the large system journey, where you're looking to automate and run different experiments and workflows, such as maybe cell culture, ELISA, high-throughput screening, for example, all on the same platform and maybe in parallel. So I would say the recent brilliant example of the lab automation success journey has been the automated amplitude workflow for our TaqPath SARS-CoV-2 assay for detecting COVID at the population scale. So as you can see that as we move from left to right, the systems get more configurable, the requirements get more complex, the systems get more advanced, and the functionality increases rapidly. So for automating the QuantiGene Plex assay, we chose the Thermo Scientific inspired collaborative automation platform, which brings configurable and interactive solutions to your lab. The Inspire platform has a flexible vertical orientation to save space, and right in the middle or the center of it is the Inspire is the Thermo Scientific Spinnaker XT robotic mover. It's a tall mover with a 360-degree workspace, such that all the instruments can be surrounded across it. The robotic mover has built-in barcode reading for sample tracking. It has vision-based teaching and on-the-fly healing. The key features are that it's highly intuitive. The Inspire platform is designed to work collaboratively alongside you. You can easily access instruments for offline use and just as easily return them back. In case the instrument is needed before it was returned, the software actually prompts you and alerts you to bring it back in. It's highly interactive. The unique Smart Handle technology provides at-a-glance instrument status updates and provides touch control for instrument access and control. Highly versatile. The platform is flexible design, allows saving space, and also allows you to configure the right solutions for your evolving lab needs. So the benefits are pretty much like of that of automation. It decreases hands-on time, maximizes throughput, enhances reproducible results. It's flexible to scale and evolve, and also saves space just because of its vertical structure. On this slide, we are showing a high-level graphical representation of the QuantiGene Plex assay run in an automated fashion by the Momentum software on the Inspire platform. Each step of the protocol is being automated by various instruments. So beginning from the very left, the liquid handler provides a 1:1 stamp from the input plate, and the stamped out plate is then transferred to a sealer. It is then incubated in the automated incubator Cytomat for the required amount of time. Once incubation time has been reached, the plate is retrieved and transferred to the plate peeler, followed by its movement to the plate dispenser or washer, where different reagents are dispensed on it and aspirated off. The plate is subsequently resealed, and this goes through multiple loops of incubation. And at the very end, after the end of the last incubation step, the plate is agitated on a plate shaker, and then it's transferred to the Luminex reader of your choice on the top right. And this entire process is controlled by Momentum software. So at the core of this automation is our Momentum scheduling software. It is a highly intuitive software for automating your scientific workflows in high fidelity using drag and drop functionality, requiring absolutely no coding knowledge. It is a data-driven bidirectional scheduler, which means it actively communicates with each device and logically takes the next step based on the inputs it receives. It is highly dynamic, making on-the-fly decisions to situations. Some of them are inbuilt and others can be added while you design your experiment. It is highly scalable, a single software being able to control multiple devices, multiple robots, and also simultaneously communicating with external repositories such as LIMS. It is highly capable with an ever-growing library of over 450 devices, thermo and non-thermo instruments. Here is a small glimpse of what goes on in the background between our internal groups to ensure that every protocol detail is accounted for accurately. This is the same protocol from the earlier slides, but each step has been cross-referenced against internal assay performance data to provide our customers with the confidence that they are investing in a cohesive, future-proof automated solution. When considering who to partner with, you have many options to choose from. And we, Thermo Fisher Scientific, collaborate internally to provide a cross-functional solution for unprecedented value like this example here. The automation of the QuantiGene Plex assay is a collaboration between two groups to solve our customers' challenges and support their ongoing success. This was achieved by unprecedented access to internal resources from both the QuantiGene and ProcartaPlex assay teams, as well as the lab automation development teams, to produce a solution that satisfied not only their assay performance characteristics, but also their evolving needs. So herein, 3 solutions were devised in collaboration with the QuantiGene team, Procarta team, and lab automation group. And these 3 options represent the level of scalability and flexibility. The platforms for all 3 solutions are Inspire automation platform based. That means they're vertically integrated for saving space, and access to all the instruments in an offline mode is easy to manage. The one on the left, the single Cytomat system on the left, it provides self-contained protocol automation for QuantiGene and ProcartaPlex assay both, wherein the input sample plate is just passed into the system, and finally the data is automatically uploaded to your LIMS when the run is complete. In the middle of the screen is the dual Cytomat system built based on the first one, with the only addition of an additional Cytomat incubator, allowing both incubators to be set at static temperatures for a considerable increase in throughput. Finally, the one on the right, the dual high-capacity Cytomat system, it leverages on the side axis of the Inspire platform. It has a slightly larger footprint for increased capacity, and with the addition of carousel, which provides increased ambient storage and which translates to increased walkaway time. So this slide shows different options highlighting the flexibility to scale and your throughput requirements. If you'd like to learn more about what Stefan and I have presented today, please do visit the links thermofisher.com/luminex and thermofisher.com/inspire to learn more. And with that, we would like to thank you all for your time and are ready to take any questions from the audience now.
And I want to thank you both, Dr. Jellbauer and Dr. Suri, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in from our audience. Our first question, how many targets can I look at simultaneously?
Yeah, I can take that question. So for both our QuantiGene Plex and our ProcartaPlex assay, you can really measure 80 different targets per well, and that is both in the 96 and 3D4 well format. And as mentioned earlier, there is different off-the-shelf panels available for different areas of biology and a lot of different immunology areas for ProcartaPlex. And then on the QuantiGene Plex side, it's really a lot of flexibility. All we need to know is the up to 80 targets that you want to measure, and we can custom build that assay for you. But as also mentioned, we are about to launch a lot of different off-the-shelf and pre-designed panels for QuantiGene Plex as well, so stay tuned.
Thank you so much. And what is the sensitivity of the QuantiGene Plex assay?
Yeah, great question. I haven't been able to talk about the details of the assay, but thanks for asking. Good question. So the sensitivity is really dependent on the individual expression rate of your targets. A general threshold that we usually put out is there needs to be 1,000 copies in your sample of a particular target so that the assay can detect it. So either you have 1,000 cells with one copy of your target that's expressed, or you have one single cell with 1,000 copies of that specific target, and the assay will be able to give you a reliable signal.
Thank you so much. And I want to thank our audience for these great questions. Our next audience member says, I'm doing a large cell culture screen with thousands of compounds. How do I do my sample prep for QuantiGene Plex analysis?
Yeah, I can take that one as well. It's still QuantiGene Plex related. So while certainly our lab automation group could help you with setting up the sample prep in terms of the steps and automating the steps for lysis, incubation, and then eventually the RNA extraction if necessary, the sample prep procedure for QuantiGene Plex in general is very straightforward. We offer sample prep solutions for both tissues as well as cells, together with protocols for different tissue types as well as for different cell types. On top of that, we also have solutions for blood samples. And I also want to mention it is also possible if you already have the RNA extracted, you can run that purified RNA directly in the assay as well. But again, the big benefit is you do not have to extract the RNA. You can go straight from your cell lysates into your assay, and the cell screening or cell culture screening is the biggest tool for high throughput assays. So in a hypothetical scenario, you would end your screening experiments. You would take off your supernatant, maybe wash the cells a couple times, and then add the lysis buffer, then incubate the samples for like 30 minutes at a certain temperature, and then you're ready to have your lysate ready for the assay. So you go straight into the assay from there. Sorry, that was a very detailed answer, but I hope that was helpful.
Of course. We have time for a couple of more questions. For people just starting out on an automation journey, what do you suggest is the first step?
Okay. I can take that question. Thank you for that question. It's a nice question. So I'd probably like to answer that with two perspectives. So at an organization level, the first step would be to identify where you would like to start off in your automation journey, and also look ahead as to where would you like to reach, and then maybe engage in a conversation. But at a more lab-centric level, a good way would be to identify either critical experiments or steps which will benefit by automation, and based on that, start a conversation with one of our sales or application scientists, and they'll be able to identify the granular details of your journey better. Yeah. I hope that answers.
Yes, it does. Thank you so much. Now, you listed a number of instruments placed on the Inspire platform. Is it possible to use the different instruments than the ones that were mentioned? For example, using a different plate reader or a plate sealer or peeler?
Yes, for sure. So the Inspire platform is highly configurable and flexible. So for this particular assay, we chose these instruments based on what worked best. But yes, you can, like a customer can choose any instrument, and we are brand agnostic. So it doesn't matter which peeler or sealer or which reader you'd like to use. Any and every instrument, as long as it's automatable, can be added onto the Inspire platform.
Thank you both so much. I want to thank you for your time today. Would you like to provide any closing remarks for our audience before we go?
Yeah, thanks for having us. Thanks for Luminex to invite us to speak on their behalf at this great event. I hope you all enjoyed our presentation and learned something new from us today. Please get in touch if you have any further questions. And I just want to emphasize again that this project that we presented today was a great collaboration internally at Thermo Fisher Scientific between our assay developer group and our lab automation group, where we work together to come up with solutions for our customers to run the assay in a higher throughput, eventually get to the data earlier and move their lead candidates further in the drug development pipeline, which hopefully benefits all of us at some point.
Well, I want to thank you both, Dr. Jellbauer and Dr. Suri, for your time today and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Luminex, a DSOR company, for sponsoring today's webinar. And questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until May 18th, 2024. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thank you.
Thank you.